Good day to you from Inside Drug Discovery. This video describes the discovery program of bexicaserin, a compound in development for treatment of seizure disorders. The program is summarized based on a Drug Annotation article from the Journal of Medicinal Chemistry. The research history is somewhat complicated and involves a number of companies past and present: Arena, Beacon, Longboard, Eurofins Beacon, and Lundbeck. A link to the article can be found in the video description.
We can start with the drug target, the 5-HT receptor. 5-HT is serotonin. These receptors are part of the GPCR superfamily, and there are multiple subtypes of 5-HT receptors. Many drugs target 5-HT receptors. For example, sumatriptan primarily targets 5-HT1B-R, and thioridazine primarily targets 5-HT2C-R. Achieving selectivity within the serotonin family, however, is very challenging and critical for minimizing safety risks. The goal of this program is to achieve high 5-HT2CR selectivity in order to treat seizure disorders, including developmental and epileptic encephalopathy. 5-HT2CR ligands, specifically agonists, are known. Lorcaserin was a drug, although no longer in the market, for weight loss. It was developed by Arena Pharmaceuticals. Lorcaserin showed moderate selectivity for 2C over 2A and 2B, and selectivity over 2A and 2B is the main concern. Arena continued working in this area. The work was in collaboration with Beacon Pharmaceuticals. The two companies worked together and reported the lead compound in the middle of the slide with much higher 5-HT2CR selectivity. The lead has a distinctive 5, 6, 7 ring system. The program for bexicaserin started with this lead. For comparison, we have serotonin on the right. Serotonin is the endogenous ligand for 5-HT receptors. Serotonin shows very poor selectivity across the 2A, 2B, 2C group. Scientists with Arena and Beacon explored secondary amide groups in the 8 position of the 5, 6, 7 ring scaffold. While not explicitly called out by the authors, a modeled structure of an amide ligand bound to 5-HT2CR in the paper shows a likely key hydrogen bond with the amide NH. Therefore, the secondary amide was a critical feature in the substituents. The scientists looked at a lot of amides. Many of the best are on this slide. As you can see, selectivity for 2C over 2A and 2B is far improved over the original lead with mostly reasonable potency. Interestingly, all compounds showed some degree of superagonism. Several of these compounds were advanced into in vivo studies. Among others, 7B, 7I, and 7L were studied in rats with an oral dose of 10 mg per kg. Food intake at 1 hour was monitored as a surrogate for 5-HT2CR activity. The researchers also looked at plasma and brain levels. Compound 7B stood out despite its relatively modest 5-HT2CR potency. This limited data set does perhaps show a link between efflux ratio and brain penetration, which would not be surprising. So compound 7B emerged as a very attractive compound. It has high selectivity, provides good oral exposure, shows promising efficacy, has good safety, but the predicted human half-life was only 3 hours. The team had a goal of once or twice per day administration, and so therefore additional optimization was needed. Here is the plan for improving the PK properties of 7B. 7B has a low plasma protein binding with an unbound fraction of 0.87 in the rat. The compound also has a quite low C log P. Based on the original ARENA Beacon lead, the team knew that substitution of small alkyl groups at the 7 position were well tolerated. So the idea was to add small alkyl groups. These would incrementally raise lipophilicity and therefore raise nonspecific plasma protein binding and reduce the unbound fraction. These changes should reduce clearance and raise half-life. Okay, here are some of the changes. Note that tweaks around the 7 position, the R' group, were explored in conjunction with small changes to the R group on the amide nitrogen. We have 7B for comparison. In the other 3 compounds, gains in potency were realized. Both 19A and 19M had similar efficacy to 7B in terms of reduced food intake. Compound 19 really shined in terms of brain exposures relative to plasma. Note that 19M also had the lowest efflux ratio of the new analogs in this table. Introduction of an R' group does add a stereocenter. The assignment of this stereocenter is not fully confirmed in the paper, but the configuration shown is indicated in the text. On this slide are the PK properties of 19M. Nothing is critical here. I will note that the oral bioavailability is good. From these data, the team predicted a human half-life of 7 hours. That is a good improvement over the 3 hours in 7B. The predicted human half-life was acceptable and within expectations for a once or twice per day oral regimen for the final drug. To summarize, 19M, also called LP352, the LP is for Longboard Pharmaceuticals, and the international non-proprietary name is bexicaserin. The compound can be formulated as a stable hydrochloride salt. Solubility of the salt is very favorable. The compound shows no identifiable safety risks in vitro. Bexicaserin is currently being studied in the clinic and has already advanced through early trials. On the screen is information for one current phase 3 trial for developmental and epileptic encephalopathy. That concludes the discovery story for bexicaserin. Bexicaserin is an orally delivered agonist of 5-HT2C receptor with promising activity for certain seizure-related indications. Thank you for watching to the end of this video. Your comments and questions are always welcome. Please like and subscribe. Take care.